good.
Hello, everybody. We're the Microbiology Journal
Morning.
Club. Morning. I'm Fazal Lam, and this is my colleague Danny Chan, and we're going to here to talk about microbiology. Uh,
Yep, very interesting topic, regardless of the circumstances of the world, but made specifically poignant by the circumstances of the world these days.
yeah. So, while I'd usually be a shut-in talking about bacteria people, suddenly people want to listen to me, which is great.
Yeah, that's the hope. Yeah, both Faz and I went to graduate school to study infectious disease, microbiology, right? You
Yep,
did
that's
microbiology.
right. But
And yeah, but we're sort of directing our attention to microbiology papers. Oh, we used to do a journal club during grad school called the Twitter Journal Club, where we had discussions online.
yeah,
It was a fun thing to do,
yeah.
and we're rebooting that in this age to talk about what we think might be topical, or what interests us in the emerging literature around SARS-CoV-2 at the moment,
Yeah.
but hopefully other things in the future.
Oh, yeah. I mean, this is a brilliant opportunity for us to actually talk about the process of peer review and papers and kind of take you through how our process for reviewing papers. And
Hmm.
and this part of so we have a week off and week on session. So this is our week off where we select our papers and talk a little bit about the microbiology news of the been happening over the past few like week or so. And then we choose which paper we want to focus in on, and then we go away and we read, and hopefully you can read along as well, and then we come back and discuss it next week, and we can discuss exactly how the science was done and how conclusions were arrived at, and hopefully it help us understand how this scientific process works.
Yeah. Yeah, and this is something that happens inside of like academic training, right? Like when we were grad students, they make you go to a bunch of journal clubs and then you start going to them because you need to learn in certain disciplines. So we're hoping to loop more people into that sort of process.
Yeah, because a journal paper isn't just like a fact that's laid down in stone. It's the start of a discussion between
Mhm.
scientists, and hopefully, if we open it up and show you exactly what we're looking at and how we try to put ourselves in the shoes of scientists who are doing these papers, understand their logic, and see what's going on, that
Mhm.
we can like help enrich the conversation.
Absolutely. Yeah. I mean, that's our hope.
That's the hope. But
The hope.
but you know,
Um, So yeah, actually, I was bringing up just before we started the stream that I found a website John Hopkins University put out. They're calling it what are they calling it? The NCRC. I think that's like, and it's a database of all the SARS-CoV-2 papers they brought together. Um, and I guess what's interesting is that they write short like descriptions, sort of like what do we learn, what's going on. So I went through that database this week a little bit with my own interest, right? There's
yeah.
there's too many to go through like comprehensively. I just went like, what am I interested in? Poked around over here and fetched some from there. Um, can I bring one up now? Is that a good time?
Yeah, sure. Bring one up. Let's hear about what caught your eye.
I saw something, Wu et al. It's called Neutralizing Antibody Responses to SARS-CoV-2. We have to figure out a better way of sharing these to each other as we go. Well, okay, wait. We have one way, or that's the
Zotero.
Zotero.
Yes.
Yeah. Um,
Yeah.
but it's this so neutralizing antibody responses to SARS-CoV-2 in a COVID-19 recovered patient cohort and their implications. It's a preprint
Yep.
from medRxiv, so that's
Ah.
the doctor's bioarchive. And in it, they look at a bunch of people who were hospitalized,
Mm.
patients with symptoms, and then they screened the serum to see whether it was neutralizing or not.
Now,
And this was something we talked about last week, right? Like when
yeah.
we were when we were studying or when we were talking about the DNA vaccine and how it was so sad there was no like functional, even like a very
Yeah.
simple functional test.
Something
So
like
here
that.
they actually do that functional test in people. Um, I don't think they get too deep into the mechanism of like what's going on, but they do, yeah, they just test the serum against a whole bunch of different things, trying to find out, what makes for a good antibodies. Uh, what might the characteristics of patients that have good antibody response look like? I think that's probably something else they're looking at.
Yeah.
It's a lot of figures. Should I send you the link? Is that easiest?
I mean, sure, you can
Yeah.
send out the link to me, but I mean, it sounds like an interesting paper. There's something we would want to talk about, I think, because
Yeah.
again, that's an important part of what we're. The whole thing is like, how do people become immune to this disease, and what happens with the immune response? Um,
Yeah. And that's like a huge, like, from the very beginning, that's been my like topic of interest, right? Because when we talk about um having people, and how about this? When we talk about ending social distancing,
yes,
right? I think that that's like, it's so important to know like what that entails and like what data do we need to know that? And like, I mean, this is not going to get us there because, we're not even talking about transmission
mm,
with these antibodies, but it's still like a step in that direction, talking about what are the antibodies doing exactly, right? Like what worth is it oh, actually two of my friends got tested, antibody
oh
tested this
really?
week.
Wow.
Yeah, both negative,
Oh,
but
congrats, I guess.
I don't know. I have no idea. That's the funny thing,
Yeah.
right? It's like both negative. We're not really sure how to interpret this information because, like, what new decisions can they make with that information in their court,
Yeah.
right? We just don't know enough about what these antibody tests are like helping us inform.
I
Um,
mean, actually, there's a paper that I came across basically this morning that was released by full disclosure, one of my like co-authors from a long time ago,
oh,
literally that's
nice.
on like systemic and mucosal antibody secretion specific to mild versus severe COVID-19. So basically looked at
Ah,
like
cool.
mucosal antibodies. So that's the antibodies that are produced by your saliva and I think blood
IgA.
IgA basically and
IgA.
IgG, and they checked and they saw. I think they looked to see whether there are distinct responses between like people that suffered from severe COVID-19 and people who had not suffered from the milder version. They found some differences. Some again, I only saw it this morning, but it seemed quite interesting that picking apart those differences because apparently for milder cases it tends to be a bit more of a trend in certain areas. The antibody response is somewhat more transient, so that might
Huh.
affect how it appears on antibody tests. So
Yeah, like if you're looking for certain antibodies in a test, right, and it's only a short window, then like it matters when you get that test.
yeah,
Yeah.
so I mean the question is like long-term antibodies and like how long the immune response lasts. That's like
Right. Uh,
yeah.
do you have the DOI for that? Can I type
I
that? Can I enter that into Zotero?
Uh, it's on bioRxiv. Let me see if I can put up for you. So the DOI is 10.1101 slash 2020
Oh gosh, I was thinking
dot.
you could just like type it
Oh
into
yeah, right.
the.
I'll just type it in the Skype chat.
I just want to, I'm just like, so we are archive, right? Like, as we talk about these things, we're trying to keep a record of the stuff that we've talked about. And it also helps us like inform, like for other people who join in, they can see like a list of things that we thought about, right? A list of papers that we may have considered.
Yeah,
People can add to that list. You can tweet us, to add to the list. Yeah, I'm just trying to keep it a bit organized in some ways, rudimentary. I mean, I'm not a heavy researcher in this field, so it doesn't have to be some advanced thing, but I think a record is nice
yeah.
in that sense.
And that
Yeah,
it's good because again it helps everyone participate in the conversation because even if we don't talk about these papers, some one of you listeners or viewers might actually want to check them out. And
absolutely.
yeah, so I mean we're going to be hovering over and basically picking out some nice juicy papers that we would like to talk about but might not have time because sometimes yeah, with because again especially with the sheer amount of papers that are coming out, it is very hard to find time to read them all.
Yeah, there are so many. It's um it's kind of crazy. I was just saying that on This Week in Virology, they said that there's so many papers, people were considering looking through them with AI, which was a research topic before. I feel like it's made like more, yeah.
I mean,
Oh, this DOI doesn't bring me
oh
anywhere.
god, oh good grief. I'll see if I can.
Did you copy and paste it or did you
I
type it?
copied and pasted it.
Hmm.
So
I'll just search it on the net. Maybe I'll come up with something.
yeah,
So DOIs are these.
dig,
We discussed
yeah,
DOIs. They're like, I guess it's an index, right?
yeah,
They're unique identifiers to because I guess URLs can change, right? When you're like revamping your
Yeah,
website, you could like add a new section, you could move things around. DOIs are supposed to always like point directly to that website
yeah,
always.
they're run by like an archiving company that I think they index so they always try to find like at least one copy of the paper that's available. I don't know exactly the because I know that there's like lots of archiving that goes. So even if like a journal goes disappears or if like a website disappears, there's always going to be a copy saved by the people who run digital object identifier.org.
Object identifier.
Yeah,
That's what the DOI
yeah.
stands for, right?
So they're there to like try and preserve the scientific record, or even like any academic record, so that
Yeah. And also just like so people can always get yeah, to get to the papers that they need. Okay, yeah, I found it when I searched for it.
yeah,
Um,
um,
I guess it's so new, it's not put into the it won't redirect properly
yeah.
yet.
So it's like came out yesterday, um, so that's why it's pretty new. And yeah, it's interesting because I mean one of the concerns about this is if they because if it because I mean there are some people who already have this kind of people are already starting to politicize things to do with COVID-19, like whether antibody tests work or not. And I feel like
Mhm.
kind of this is one of those papers where the nuance of it will be lost if we don't like look into it directly. But
Sure.
uh,
I mean, there's like so much nuance to antibodies in general. Well, like when people say antibodies, right? You think, oh, antibodies, those Y-shaped. I mean, I would assume people think Y-shaped
yeah,
things that your body makes that like are essentially bullets. I feel like people think of it in terms of war.
yeah. I mean
Like
they're a cool thing.
bullet against.
Whenever I want to like teach kids about it, I always say, yeah, antibodies. They're like bullets, they're like machine guns. You see here, germ, and then yeah, it's an entertaining way of thinking about it. And I mean, the another thing, like same with antibodies, camelid antibodies have been something that have been. I mean, there's some. So I think there's like they're smaller antibodies with camelids,
Yes.
and they're
The product they ended up calling nanobodies,
yeah
right? Or something like
nanobodies.
that.
So these antibodies that come from llamas or alpacas, they alpaca punch. But yeah, I mean, there's something that people have been interested in for a while because I think having them small makes them more druggable. So they're a bit
Right.
easier for people to actually make in a lab, and they can get to all sorts of places that normal antibodies might not be able to get to. So there's a lot of
Right. And all that being said, it's like so antibody really doesn't mean like a single type of
mm,
molecule that this shape that your body makes. There's like a diversity of antibodies in your own body, but as well between species,
yeah.
right? And like like all of those have different potential applications just depending on how you figure it all out.
Yeah, I mean, I have colleagues who are working with with egg antibodies, which were had a special because there are some super antigen, so some like for a specific function they work very well for. I don't want to go straight into the project,
Yeah,
but basically they're hitting a problem where their experiments weren't working, so they went into egg antibodies and they did work because they're structured differently, so they didn't so they could do certain things.
sure.
I mean, there's
No one knew about egg antibodies. It was causing them problems in their experiment, so they had to study egg
yeah,
antibodies.
people we do get pushed down that route. I mean, I had to learn a bit of physics during my PhD because I was working on bioluminescence. That means I had to learn
Hmm,
about luminescence in order to figure out how to interpret, say, a luminescent signal into bacterial numbers. You get pushed down to all
sure.
sorts of routes when you're doing your PhD, and basically there isn't a set syllabus. You just find a problem, you go, I need to solve this, and I need to figure out how.
Yes. Yes. You try to learn what you need to support that. Okay, I added that one to the our I added that to our list too.
Yeah.
Yeah, well let's go back and forth. So then that was the most recent one that I found. But then I also is this a recent one? Oh yeah, I also found a recent paper. This is structural and functional basis of SARS-CoV-2 entry by using human ACE2. This is
Ooh.
published, I think, in Cell.
Yeah.
It's a big paper. Um, and I think in this one they actually have a neutralizing antibody that they found, and they co-crystallize it, or they're actually specifically looking at its binding. Um,
Ooh.
oh yeah, they co-crystallize, or I don't co-crystallize, but they 2.5 angstrom structure, meaning that's like a resolution, very
Yeah.
small.
So that's very small. That's like on the atom level. So looking at
Yeah,
atom by atom,
atom by
these
atom. How does this one antibody touch the receptor? Yeah, the
Yeah, so
SARS-CoV-2 binding domain.
yeah, this is those because I love these. So they basically
Ah,
like kind of get all these things to join, and then they like freeze them in that moment, and they look at how both of these proteins are interacting. So you get these really
yeah.
fancy 3D images.
Oh, for some reason I thought it was. Oh yeah, I see. They did do antibodies, but the main thing they're reporting is actually how it's touching the receptor, right?
Mm,
ACE2. Yeah. So I mean that's like this is like from the biology side, not from,
yeah,
like understanding like how does this actually work like at the atom level.
yeah.
But
Looking
again,
at
knowing what those contacts are can tell us like if an antibody was directly competing with those same contacts, it would be a really good antibody, right? Like that's kind of what we're getting at in this study. Yeah, so this one's published in Cell. It was published, what date was it published? On the 14th of of May. So
yeah,
it's quite a recent publication. Yeah, because I mean, I did look at a few. I guess I'll throw this one on too. That I also picked. I don't know if we should talk the cryo-EM structure of the CoV-2 spike protein. We actually talked about it recently, like in the last paper that we
yeah,
covered. They said that it came out while they were while they were developing their vaccine candidate, and then they decided based on that to go with one of the candidates over the other. Um, but yeah, that would be something I guess about the structural biology. Like we didn't
yeah,
do that when we were in school, but but it is information that we'd be able to interpret at some level. And again, like you have to go down these paths if you're trying to understand certain
yeah,
things. Yeah, you have to know what you can get out of it.
yeah. And also like there's been a few vaccine papers that have come out this well week.
Mm.
So there's been so I know earlier this week I think I tweeted you about a paper on like the Chadox one vaccine
Oh
in
yeah,
rhesus macaques.
it was published this now. Is that
It was
what's
just
happening?
published.
Yeah,
Oh yeah, it was so it
because
was a
I remember
preprint.
we had a preprint a while back.
Uh, so this is of the Oxford study,
Mm.
and like it hit the news fairly recently. But it basically with there are two sets of interpretations that came out. One that it works in that the macaques showed better pathology results, or like they seem to have less severe symptoms.
Yeah.
And the other one was the other interpretation was, oh, it still produced virus. They were still infected with the virus, and
Ah,
so there is a. It's one of those papers where there is a fair bit of nuance, and suddenly once the general discourse caught its hand on it, it like kind of split apart. Whereas I got really extremely like,
sure. Some people
okay,
said this is never going to work. Some people said this is promising for X, Y, and Z reasons.
yeah. I
Uh,
mean, I mean, it looks relatively promising. I haven't looked at it in too much depth. I mean, I think based on the Lost episode, where we're looking at pathology, my trust of pathology has like been shaken quite a lot.
sure.
Um, but I mean, again, it's better than nothing.
Right.
But and the second vaccine paper was I think the first human yeah, the first human trial of like safety tolerability of an adenovirus type vectored COVID-19 vaccine in an open label non-randomized first in human trial.
Hmm.
Um, so that's
Wait,
an
what vaccine is that? Sorry,
so this was a vaccine based in China from China from that's based on adenovirus, but not
yeah, the
the
chimpanzee. Yes.
chimpan. Yeah, so it's a so yeah, this one kind of like I missed this one for a bit. I mean, again, another big vaccine news was the other big RNA vaccine study, which they released a press release about it saying
Yes, I
it
saw
looks
that
good.
the press release, but not. And that I did actually. I added one of those to the list because there's the press release. I mean, we can't talk about a press release. There's no data in press releases. No data to
Exactly.
criticize, at least. Just tables of promising numbers.
Yeah.
But that did make me go, and I did add this also to the list. I went in to get respiratory syncytial virus.
Mm.
This platform has been used to deliver vaccines in a bunch of different circumstances, and they have better published data for their results with respiratory syncytial virus. So not again, not
Hmm.
directly relevant, but it's using the same technology from the same company. They had a publication the 2nd of or the 14th of February, 2020,
Hmm.
with respiratory syncytial virus. Yeah, this is a mRNA virus, Moderna.
Yeah.
I think people are saying this is one of the farthest ones along in terms of development.
Uh, I mean,
Lana?
it wouldn't surprise me, but because I think there was a lot of effort thrown behind it.
Yeah.
Um, but again, like the first one that seems to have gotten the published study is this. Well, because I remember there was one Picovac that might have been published, but I didn't really haven't been keeping up with.
Yes. Actually, I picked a paper from that one too. That's the inactivated vaccine,
Yes.
right? Yeah. Yes. Yes, that was published May 6th. I picked that one up as well. That's also an interesting one. Yeah, an inactivated vaccine candidate for SARS-CoV-2. I didn't realize that it was published. I was like, searching through. I think I got through this from that John Hopkins website, which was quite useful.
Yeah.
This
Yeah. We.
doesn't have any. Did I pick up like the perspective? No, this is it.
Oh.
I just can't. It's funny. There's no figures. There's no figures in the version that I picked. Oh no, they're all back here. I see them. Yeah,
So.
yeah,
Yeah.
inactivated vaccine. So this is like the class. This Picovac. Yeah,
Yeah.
this is what you're talking about. This is the classic way of making vaccines.
Yeah. The
Yeah,
classic
from the
old
beginning.
school.
Weak virus. Or actually, my professor in virology used to cite wimpy virus.
Yeah. I. Yeah.
I
Wimpy
don't know
virus
why
is.
wimpy was a better word than weak, but it just made me giggle all the time, like the wimpy viruses.
Ooh, I can't handle your immune system. I'm too wimpy. And the immune system, right? I'm gonna profile you, and all your cousins are coming here.
Exactly,
So
exactly.
when the Chad version comes in, it's like, okay, fine, you're done. It's like kind of the snitch version of viruses, like
Totally, yeah.
giving away. Yeah, that's
Yeah, so
that's a
that's published. Yeah, we've been talking about preprints this whole time. I feel like it'd be nice to do a paper that was actually published.
yeah. So that paper has actually been published. So that is a good one to look at.
Yeah, so
Um,
that's another candidate. Okay.
uh, what are the ones? Because uh, I'm thinking of whether because I think those are the vaccine papers I saw, but I did see one interesting one about concerns about the immune response to SARS.
Mm,
So there's one that was published in Cell again. Imbalanced host response to SARS-CoV-2 drives the development of COVID-19, which which again seemed like an interesting one because it kind of indicated that it was slightly doing something slightly different than what you'd expect from a coronavirus.
hmm.
That it has a nice graphical abstract, which is
Can you
always nice when
can you
people put
drop the
the.
link in the chat for me?
Yeah, sure. I'll drop the link in.
Link or
Uh,
DOI, whatever is easiest. DOI is
uh,
easiest for me.
yeah. I'll see if I can find the DOI for it.
Oh, but if it's too difficult, then the link is fine.
Yeah.
Okay, I got it.
But yeah. So, it's essentially saying that how like SARS-CoV-2 is become how does it become more severe,
Mm.
or and how does it act different? So
Yeah,
they
are you saying like between the two paths, right? Because like everyone reacts different, or like it seems like from what we see in the clinical course, people are reacting differently. Some people are having like severe disease, some people are mild, some people asymptomatic, right? And so there's some underlying host understanding there that might tell us what's going on.
I
Yeah,
mean, it might give us some information on that.
yeah.
Uh, I mean, I think it's mostly focusing. It's very immunology like uh,
Flow
focused paper.
cytometry graphs, lots of bar charts.
Yeah, lots of like different cytokines, and there are so many cytokines, and immunology is almost like it's as complicated as neurology because there are so many different information networks that pass around. So you have to.
Totally.
So when you so
I
when you're
mean,
looking at you,
the job of the immune system is this very tricky thing of like what is dangerous versus what isn't dangerous. Because if you're
yeah,
recognizing everything, then you're just going to be in a state of constant inflammation.
yeah,
Um,
it has to communicate. It has to have a memory. It has to do all sorts of interesting things to try and
yeah,
combat diseases. and to not accidentally destroy you. So it's
Yeah,
actually there's a lot of intelligence in actually mapping out how it's to borrow a phrase, how its internal thought process occurs when it's facing
sure,
disease.
sure. Oh yeah,
So
this
yeah,
one's very recent as well, May 15th.
yeah. So I mean, it's interesting, but I feel like I would need to literally talk to an immunologist about this
Oh, very
it,
dense.
because they've volcano plots, and volcano plots are like really, they're like they're graphs that I don't quite understand yet, so I need
Mhm.
to learn because they're
Oh
from
yeah,
like various gene correlation again, because again they're looking at so much data in one go. They need to be able to
I see.
visualize it and to
Yeah, this is
look at
immunology in the age of omics.
yes. Uh, which is much more interesting than when they said, "Oh, we found a new cytokine that's or a new type of cell that we're going to call a B helper cell or the Th5." I mean,
I mean, everything has its use, right? Like, I definitely think that yeah, there's this thing in immunology where it's like you find the new subsection of your favorite cell type, right? And
yeah,
see that that new subsection does something very unique in a very unique circumstance.
yeah.
Um,
I mean, I
but
feel
yeah,
like
that
that's
you can
like
zoom out and also look at the bigger picture. Is that what I guess the omics is trying to do?
yeah, and again, I like the idea that because I think I while I was in undergrad, there were people who were saying like we want to move beyond that to the this idea that like but again it was very complicated so I didn't really pay as much attention as I needed to
Sure.
it, but moving away from thinking of like specific cell types but as in for like kind of a cell map a kind of a space where cells can have different like reactions so
Huh.
yeah, I know. It was a well, actually, that I understood at the time, and I was really impressed that with until about 5 minutes later when all of it left my head, and I was like, because it was yeah, there was some really good graphs there, but oh.
I will say though that it's easy to get wrapped up. I felt this when I was doing school, and even now when you read papers, it's easy to get wrapped up in saying that you can describe all these broad trends using like big data and like
Yeah,
getting real deep into things. But that doesn't necessarily remove the necessity for like the small like hand scale sort of experiments. I mean, you still have to have statistical significance, but being able to control all those variables to validate maybe some of the results that are coming from the big computational things. Like there's still that value, right? Like
oh yeah,
scientists aren't about to be replaced at this point by like statistical computing machines.
I mean, although apparently we are trying to find ways to replace us with AIs to read papers for us.
Yeah.
So yeah, to creating an algorithm. I mean, to be fair, I mean, as working in a scientific journal, one of the big things we have is trying to get scientists to read papers and review them, and that is actually very, very hard.
Mhm.
And because scientists' time is being like compressed, and so some journals actually do have AI research assistants that help them read papers. So that
Interesting. Just like to score them, like a rudimentary score or
yeah,
something,
just a
like work.
rudimentary, just for basic things like scanning for plagiarism, ethical issues, or just like
Oh yeah.
phrases that might be like if it. I mean,
Red flags.
yeah, like we disprove Einstein. I'm like that's an instant red flag. I mean,
Yeah. Yeah, it's true. You gotta, you know, scientists are people too, and sometimes they
yeah,
have a chip on their shoulder, and it comes out when you're writing the paper
yeah,
that like,
and anybody can submit to a journal these days if you got like if if you got the right formatting software, you can just submit and no one can stop you, no matter how much we try.
absolutely, absolutely. Okay. Oh, I found a preprint. Oh no, this is in Science. A selection of viral variants during persistent infection of insectivorous bat cells
Oh,
with Middle East with MERS
okay.
coronavirus. So it's this is like a it seems like the study went on for a while, published now. It's they have like a cell culture of bat cells, and they passage MERS in it repeatedly for years, or I'm not sure years and years, but like for I think they said hundreds. They did hundreds of passages in bat
Oh
cells,
wow.
and then they sequence the viruses that come out, and they say how does that differ, right, from the MERS that we put in. And so this is sort of some experimental evidence to try to support these hypotheses of the emergence from bats and like why are bats the place that we're seeing a lot of coronaviruses emerge from? Can we look at some mechanistic things? Um, yeah, and they see the difference across the different genes, and I do believe that they see a difference that we see in SARS-CoV-2, the ORF5, whatever that means. Well, I'll have to read more about it.
Open reading frame five, sort of. So imagine that's like a section of the genome of MERS that does something
Right. But
specific.
what I'm saying is OR5, I think it was said also in the SARS-CoV-2 genome, right? Like what is that protein
Yeah,
exactly? And why does mutations in that region? I think it's a truncation happen as it goes through these bat cells. And I think it had some implication for infection as well, at least like a hypothesis. But again, like this is one of those things. It's like there are all these there's all these hypotheses on how it happens and like our best information to know how, but can we actually get some experimental evidence to show that this is the case? And this would be a paper like that. Yeah, experimental
yeah,
evolution, I guess, is sort of how you could characterize
yeah.
this work.
I mean, experimental evolution is one of those like those topics that has a lot of ethical kind of concerns. If you're working with like, oh, we're gonna experimentally evolve this MERS virus, and at the moment I've like horror movie kind of uh,
For,
outcomes, like where
for
like, oh, we've
sure.
created a hypervirulent version of this virus by passaging it.
Yeah, yeah. I mean, I mean, people are concerned about that sort of stuff, but again, like they're at least they're passaging it in bat, right? Bat cell culture. So like that's not gonna cause harm, presumably to human beings. But I'm sure, yeah, those concerns would also be important to talk about with this paper.
I mean, yeah, because I know it's something that I was quite worried about when because in my PhD I worked with hypervirulent version of the flesheating disease
Mm.
that that was actually like weirdly enough not as dangerous as you'd expect, but
Yeah, because it has to
is
get like
actually
into the right location to do its work, right? It's not,
yeah,
yeah, it's not going to get easily
and
into the subcutaneous.
yeah, I think it was like the thing I found in my studies it was less good at transmitting than the normal ones because it's spending all of its time being like creating all these virulence factors
Mm.
that it got all tuckered out and couldn't actually set up an infection properly unless it was already unless you'd already done all the work to put it inside
Sure.
like a severe area.
Yeah. Yeah.
But yeah, I mean again, this is it's an interesting paper. I mean, yeah, those kind of structured evolution papers are interesting.
Yeah, yeah. I guess you can see that like I was pulling in some ways I pulled away a little bit from looking at direct SARS-CoV-2 stuff just because there is so much that supports what we know about it as well.
Yeah,
I mean it takes time to sort of understand that. So it might be interesting to tell a story from I mean this is still published recently, April 29th, 2020. You know, it's obviously come out in during this era, right? And like accelerated through the process because it might have something to tell us about it, but it's also not it's not like directly related to a therapeutics thought yet. I mean, yet, like
mm,
that people can think of, right? That's one of the things about science. I mean, I'm sure you experienced this, like the difference between basic and translational work,
yeah,
and like what's the value of basic work, basic research. I mean,
yeah. I mean, I was mostly a translational scientist, so basic research where the people we nicked our results off and been like, okay, now does it work in a rat? Does it work in an animal? Does it work in a human?
sure, yeah,
Does it?
yeah, like
So a
getting
lot of
closer to the therapeutic side of things.
yeah. For the buzzword we had was bench to bedside. So we found this interesting
Yes.
thing on the bench.
I-
How does it translate?
Yes, absolutely. But at the same
Whereas
time, like
like
bench to bedside, you're still just using rats and guinea pigs. So it's like
yeah.
the
So
journey from
yeah,
bench to bedside is extremely long, and no one person could encapsulate that transition.
yeah,
Yeah,
exactly. And it's less into like finding out the answers of how things work. It's more into finding out like whether they work or and more looking at you end up answering different questions to what you like. You end up finding out a lot more about the difference between the mouse and guinea pig and human immune systems, and you learn about say how like a virus works.
sure.
There's a lot of like in, interesting aspects. You don't look at a basic science, or you there's all sorts of interfaces that happen. They're all important,
Yeah.
and that results in this like entire deluge of research that we're trying to like kind of struggle to keep a track of.
Yeah, because everything falls on a spectrum, I guess, right? Between the bench and the bedside is like a good way to explain it, I guess. And that like there's always a push, especially with funding bodies, right, to go towards the bedside,
Yeah,
um, which makes a lot of sense because we want to be spending money there. But then there's also this thing. It's like, but that research is like a continuum, you know, like you
Yeah.
need the people to like start the ideas here and evolve them and go in different ways. So
Yeah.
yeah, it's all good
I
research.
mean,
Well,
yeah, I mean, because having like basic understanding science is so important because at the other end, I mean, we're seeing it a lot like in papers coming out about COVID, is that some of it is just throwing stuff against the wall and seeing whether it sticks or not. So
yeah.
things like hydroxychloroquine and a lot of like antivirus people testing, they're like things that worked in that might have been working in other viruses that is testing out on COVID too to see whether they work or not.
Right. And if we had like, so I guess the thought would be like, let's say we had a more developed knowledge of coronavirus pathogenesis, right? It's possible that we could exclude some of those things based on our well-established understanding. But you know, we're not there, so like
Yeah,
we're doing everything along
I
the
mean,
path.
yeah, we are in the middle of an emergency, so throwing stuff against the wall to see whether it works is actually an acceptable response, rather than waiting to find out. But how does it work? What matters is it works. That's true.
Right, right, right. But knowing, having a good concept of how things work could inform like better, right? Like you could come to a better reasoned guess, right?
Yeah.
Better reasoned hypotheses to like why to go down certain paths versus others.
I mean,
Yeah, different information for different circumstances, right? Different levels of evidence too for different purposes. I think that's also important to remember.
yeah, I think that this has like been come to a head with the hydroxychloroquine sort of thing because that's one of those things
Mhm.
that was actually it works kind of it might work so let's just try it anyway to see whether it works or not. And
Yeah.
then there's a
And
big
then they did all that work in humans and
yeah, there was a
yeah.
big study that came out recently that states that oh it might not work and it might also cause some nasty side effects and actually
Yeah.
reduce like life reduce patient outcomes, which is
Mm.
another interesting paper that we could potentially take a look at.
Huh. Yeah, I saw in the news there was also word that the larger trial of remdesivir that they were doing that has been prematurely terminated because of positive results,
Yeah,
right? But there was also talk in that article that I read that, you know, it's unfortunate that we're in the age where we have to terminate because of these positive results like so quickly because there could be, you know, like there might be things that we could have learned if we had better separation of the data between the placebo arm and the non. But it was just like, you know, it's a decision made by doctors, which, you know, importantly, doctors
mm,
should be making those decisions that
yeah,
they see benefit. So, like, let's give the people on placebo the thing that benefits them. But, you know, from the understanding point of view, there's something missed. You know, like it does take time to do good science. And right now we're trying to fit, like, solve the problem, answer a crisis, but yeah, but some things will take a backseat in that search.
yeah. Uh,
Do you have anything else? Did you find anything else, or should we talk about what we've taught, or should we try to choose from the list?
I think I have got one more paper. Oh yeah, on the Kawasaki-like disease that appeared in. So the in
Oh, is that the skin, or
uh
are these symptoms? Kawasaki? What's
so
I heard
yeah,
something.
it's a disease kind of kind of I think it's an autoimmune disease of some kind maybe.
Mhm.
So no, it's a self-limiting vasculitis of medium vessels. So and it primarily affects children. So it kind of basically affects blood vessels, and it
Yeah,
kind of causes an infection. And it and like they basically had this study where they kind of noticed some a kind of a group of 19 patients who were all like children aged quite young, who also like had like coronavirus who showed up with this disease. Um,
yeah.
Again, it's been published in I think it was published in the Lancet or um, I mean I'll pull up the link for you quickly to see whether I can.
Yeah, look at the
So
link.
yeah. Yeah, it was published in the Lancet in maybe a week or so ago. Um,
Okay.
Let me double check on the dates for this. Uh, so it was published on May 13th. I'll post you something. So I've got a link in the Skype. Unfortunately, it's just a plain normal link apparently.
Oh, that's fine. The links are good too. Zotero, you know, has like a nice automatic import button, so anything works really.
So
So
yeah,
this,
I mean, because again, one of the things that was fortunate, we were all hoping for coronavirus, is that they didn't affect children that
hmm
severely, and this
hmm
seems to indicate there might be something like some kind of rare complication that happens that people might want to
Yeah,
look out for.
I did hear something along these lines as well. I think it's also, and I think, yeah, you said inflammation of the
Bl-
of like small blood vessels. I think
yes.
it does end up looking like a rash maybe or something
Yeah.
like this. Yeah, which is interesting. Yeah,
So
it's kind of this is like the whole thing where it's like everyone's different. So as more people get infected, you see the interactions with different pools of the population, and there's many ways to break up. There's many ways to disrupt the human body's regular
yeah.
operation. Um, yeah,
We are complex robots.
yeah, totally.
But yeah,
Okay,
I
I'm
mean,
adding
looking at
this
it,
one.
it's a retrospective like cohort study, and there doesn't seem to be fair, like in terms of discussion, doesn't seem to be much for us to get our teeth into for it. It's just very much a,
Yeah, yeah,
re,
retrospective cohort. That's like very doctor territory.
yeah, very much a territory. So
Clinical, heavy clinical territory. Cool. Oh yeah, actually, I did have one more that I looked at, but this is from earlier, from the end of March. There might be better ones in this category
okay,
now, but it's viral kinetics and antibody responses in patients.
okay,
So like patients over time at each point. Yeah, I think we were basically talking about this when you were talking about the IgA IgG thing. Yeah, taking samples from people over time and looking, yeah, what happens in the swab, the sputum, and the stool
okay,
when you look at when you look at PCR tests. I think they also look at antibodies. Yes, they also look at IgG and IgM titers across time. Yeah, days from symptom onset, and then yeah, that moving. Yeah, so this is again like getting at like what's the course, right? What's all the different ways that you can measure things, and what's the best time even to measure? When could you potentially see something?
yeah. I mean, I feel like we did have talked quite a lot on those like antibody and immunology style papers. So I
Yes.
think that because I'm last time we did focus on vaccines, and I don't think those vaccine papers are going to go away, but I think it'll be nice to focus on something a little bit different.
Not a vaccine this time. Yeah, we talked about one that we can always go come back to a vaccine
Yeah,
one, right? Like,
I
yeah,
mean,
yeah, I agree. I like this idea of going for some variety when we're choosing. I think that would be nice. So yeah, so that means not the Pico, not the old Moderna one.
yeah.
Um,
I mean, I mean, maybe we can talk about Moderna. If they release something in the next 2 weeks, then maybe we can go in the week after. But
yeah,
yeah,
at the next one. Yeah. Um, so we have some structural stuff, right? We could talk about like the older spike protein one or the one where they co-crystallize with the receptor.
mhm,
That's like kind of on the structural side of things. Then we have some antibody stuff, right?
yeah,
Like this one, the one that you're talking about from your coworkers, right? That's like some immunology.
yeah,
I guess it's also related in some ways to the omics immunology one that you
yeah,
pointed us to. Yeah, omics immunology. And I would say, I mean, those are more on the basic research side, but it is also a bit of the same category as looking at the antibody responses in people, their serum,
mm,
and seeing the serum, how that works, right? The first one that I
yeah.
mentioned today, Med Archive.
So
So that's like three categories we have. We know we don't want to do one of them. Oh yeah, and then we have the passaging in insectivorous bat
yeah,
cells, not SARS, MERS
hmm.
evolution. That's very different than something that we've talked about. Um, yeah, that's a different section. So actually, I want to go back to that NCRC, John Hopkins School of Public Health, the database of papers that they brought together. They actually split their papers into topic sections. This is the
Yeah.
work they're doing, right? Is try to organize things for us. I'll send you the link. And the topics that they have might be helpful also to think about like choosing things from different topics
Right.
like as we go week by week,
So
not doing too much in the same
the
topic.
2019 Novel
Uh,
Coronavirus Research Compendium. Uh, I guess
yes,
I'll put the
exactly.
link in the description.
Oh, you found it.
I
This is
found
the
it, so I'm
yeah
gonna.
yeah.
th-
the topics they have is diagnostics, modeling, epidemiology, pharmaceutical interventions, non-pharmaceutical interventions, clinical presentation and prognostic risk factors, vaccines, and ecology and spillover.
Ooh, yeah, no,
Yeah.
those are the main things that that covers basically everything. And
Yeah, right.
yeah,
It sort of covers everything. And what we've said is that I feel like we've talked a lot about vaccines
yeah,
and maybe and the lost episode is pharmaceutical
yeah,
interventions, right? But there's this other side of things. We haven't really directly talked about diagnostics. We haven't talked about ecology and
yeah,
spillover. Um, and then there's stuff that like I don't know, like where does this I guess diagnostics that could be some of the immunology
yeah.
stuff we're talking about.
Because I feel
Yeah.
like that would bridge a bit of both. But I mean, and it gets
Right,
into the
right. I mean, it's also kind of vaccines because you're saying like what's useful. Like, these are very basic research. They kind of fall under, they're not about these applications
and that's
necessarily.
what makes basically research interesting because it is you have a question and you just go and say all right and your question isn't well, you don't care about the outcome, you just want an answer. And that's what I find
Right, right.
quite interesting about because I know that Lancet the omics paper the from the Lancet that seemed quite interesting to me in that regard. But
Hmm. Hmm. Yeah, they're just like they look from above and they just see what weird interactions have come out from this network of immunological data.
yeah, and what else?
Right, that was the imbalanced host response,
Yeah,
right?
imbalanced host
So
response.
SARS-CoV-2 drives development of SARS-CoV-2. Yeah, I say I also agree. I don't think we should do the Kawasaki-like disease. I'm not into that data.
Yeah, I
It's just like I'm not talk about retrospective cohort studies. It's like, okay, so like what's the patient? Yeah, and in some ways it's very similar to some when we talk about clinical studies, right?
right.
Because we'll be like, what is that patient population? Like, does it have any limitations of what it can show us?
Yeah,
And then what does it show us?
yeah,
Yeah, I want to see some pictures of cells or bar graphs with.
yeah. Give me
I
some
want to see some.
graphs. Need some graphs.
I want to see some bench stuff.
Yeah, I mean, we can
Yeah,
really.
observation
Yeah,
cohort.
otherwise it's going through a table, going, oh look, that it does this in this population. Was that statistically significant?
Yeah, I want to see some cool
Yeah,
pictures, or I want to talk about some pictures.
yeah, totally understand that. Um, yeah, I'm just going through the viral kinetics and antibody responses paper, which also that has
Oh,
lots
yeah.
of cool graphs and pictures.
Hmm.
But
Hmm.
yeah,
Yeah, it has a bunch of graphs. It was some of those graphs are very similar to the graphs we looked at last week.
yeah,
I mean, they're used differently, right? But they're the titer graphs, right? They're like we tested this range of titers against this range of the antigen.
yeah. I mean, yeah. Looking at maybe we can do something a little bit different perhaps this time, but
Yeah, I mean, we can always come back to that. I am thinking that those bat things that bat paper is significantly different.
yeah,
Um, we haven't done something on structural.
yeah.
Uh, I don't know if you feel what you feel. Rather, we do the original like structure of the receptor or the co-crystallized version. Um,
I'm quite interested in the co-crystallized version if we do that because again that kind of
yeah,
relates it to something that because again like at the beginning of this outbreak there was a lot of concern over the use of like ACE inhibitors and it's
yeah, that's right.
something that
That's right.
so
I don't know if we'll speak directly to that, but I mean, it is the data that it could it
I mean
could it could speak
it'll provide us with an opportunity to talk about that I think.
as well. Yes, yes, yeah, and I think that's another important thing to say that like there's so much going on, right? Like all these little thoughts that we might want to communicate. Yeah, will come up also in like the thematic background that we're choosing to speak in. The subthematic background. I feel like as scientists, it's like there's this big theme. It's like SARS-CoV-2, and then there's all these tiny little themes inside of it,
Yeah,
right? All these different aspects of that.
yeah, it's I mean microbiology has layers, that's why we find it so fascinating. It's
Yes, like ogres and
yes,
onions.
exactly like augurs and onions. uh, Sorry, Scotland. I've mocked your accent. Sorry, all of Scotland. Um, but yeah,
um,
uh,
yeah, I'd be happy with either. Yeah, so like I think feel like co-crystallize or bat cell passaging of the virus?
Out of those, I feel like the co-crystallization is, well. I mean, the bat cell has lots of like cool pictures. Um,
It does. It does have pictures. They're like, yeah, I think they're simple pictures, but they're
yeah.
pictures.
So I mean, it's a real toss-up for me because I feel like the co-crystallization is hitting something that I'm very interested in, but the bat passaging, it can tell us like an interesting basic science story, and it's a
Yeah.
scientific.
I mean, we haven't talked about where the virus has
Yeah,
come
we
from
haven't
yet.
really talked about that, and it's scientific reports,
Yeah.
so it means that we can really like beat this paper up if it's doing something wrong. So, I mean, it's not gonna hurt us because nobody likes scientific reports that much. So it's just, yeah, sure, it's bastard half nature. It's
Son.
fine. You can get into Nature or any other paper. Scientific reports, it's fine. Yeah.
Nature Publishing
Yeah,
Group.
Nature Publishing Group, but it's their Cascade journal.
Yeah.
It's where you get to. It's your like booby prize when you go to submit to Nature.
But for those who are not like in on right, like the scientific publishing career game, right, is that like there are these large publishers that hold so much
Yeah,
reputation for a budding researcher or even just established researchers. So
yeah, and
and they have a business model.
yeah.
So
So I mean, working in research integrity, a lot of the people I dealt with, well, they publish in a lot of different types of journals, but some of them tend to. I mean, I don't want to say anything bad about scientific reports specifically, but I mean, it's got like there. It has a diversity of different quality articles in there, which is quite refreshing if you want to look at kind of the broad scope of like how science is done. Even so, because there are lots of labs that aren't necessarily don't have all the resources, and so it's interesting to see how
yeah,
they solve
yeah,
problems as
you can
well.
only get partial answers to questions, right?
Yeah,
Like the holy grail of a scientist would be like we had a question and we answered it to the fullest extent of the technological abilities of the time.
yeah, and
Yeah.
yeah. I mean, and
But that's not always the case. Like sometimes you set out to answer things with a certain methodology, and that methodology shows you like shows you something but with heavy limitations because of like how that study was done. But that information is still important to talk about. It's still part of the
and
story.
sometimes stuff gets bounced because of genuine like bias in literature. People like do get hooked on like their specific ideas, and so if you produce something
Oh, sure.
that's like really out of the box,
Yeah.
then suddenly you end up in scientific reports, and it might be still be good, but you don't.
Right.
So
Right. Yeah, because people can't easily assess the quality of this, right?
yeah,
And say like this.
it's hard to read
Yeah.
papers, and a lot of time people will just like go with their first impressions.
Yeah, that just reminds me of something I thought a lot about in grad school. It was about like how grants are awarded, how grant review is done, right? Because it's like you're asking, someone who's very deep in what they do, right? And like being reviewed by other people in this like social scenario, right? Of
Yeah,
like people have their own biases about like methodologies and what they think is going on.
yeah. I
Um,
mean,
and then we're awarding money to people based on that, right? To do the work. It's very tricky.
yeah, and there's also you never know who's going to be on your grant committee, and
Mhm.
there's always a diversity of opinions on those, and people who argue. Just like it's like peer review, except like in person and and with much less information to go off of because you're
Yeah.
looking at like stuff that hasn't been done yet, and so it's
Yes. Yes. Yeah. Trying to choose and what I remember reading at the time and thinking, I still sort of believe this, is that like if you can't show it's difficult to show mathematically, like it's difficult to show with science, right?
mm,
That the method that we have gives us the best results, right?
yeah,
In terms of making research go. And one of the alternative models that people were thinking about is like a semi-lottery system. So it's like review is only to hit certain bars, right?
yeah.
Like review the grant application only so they hit certain bars, and then everything goes into a lottery.
I mean, I'll be honest, that almost sounds like the system we already have, but
Well, no, and now it's like a lottery based on the people who sit at the table,
yeah,
right? Because like they're still all choosing, like, right? Everyone at least how it worked in the States is that like everyone knows what number you need to be funded or not funded.
mm.
So like if you're not giving that number to people, you're basically saying don't fund. Right? So that's kind of like one of the, that's one of the things that reviewers on those committees think about is that like am I saying yes to this or am I saying no to this?
Yeah.
Yeah.
So
So yeah, there is a lottery element there, but it's also like that's a person. That's a person saying yes or
yeah,
no.
that's true. I
Yeah.
mean, it's I get because again, like those sort of grant proceedings aren't really open in the same way that science is at the. So again, we don't see the logic as much as to what
Yes.
gets grants or what
Yeah.
doesn't.
Yeah. Yeah, I mean, the part of the push for part of the push for open science is also like opening up the peer review process
Yes,
and getting transparency on these decisions as well.
I
Yeah,
mean,
with the caveat that we've now been flooded with tons of information from so many different sources.
yeah, I mean that's the kind of trade-off in open science that now we're. I mean that's why open peer review is so hard to do because you have to find people who are willing to review a paper in public, kind of like what we do, but
Right,
like for a
right.
billion papers that are being published.
Yeah, yeah. And for people who are in the career path, right? I think that's the other thing. It's like this is your for scientists who are reviewing papers for journals, like that's their chosen career,
Yeah,
right? Like their peers, right? Like if they take a shit on a paper, like that may come back to them in some way that they'd be afraid and and does that align with what is a good scientific review, right?
yeah,
Like that's like the tricky
yeah.
part.
Can you be as candid if you know that you're reviewing your boss's work? I
Yeah,
mean,
yeah, yeah, totally.
just that paper that was about like antibody responses, IG. I mean, again, that's like one of my old co-authors who I've never met in person, but I just don't want to be like,
Mhm.
oh, I've hurt her feelings. Oh dear. Whoops, that's one
Yeah,
person I'm
yeah.
off the Christmas card list forever. But I mean, that one stopped me.
Okay.
But I mean,
Yeah, I mean, like ideally it shouldn't, right? But
I mean, well,
we're not perfect robot
I
people.
mean, that's the kind of advantage of the fact that we're outside of science a little bit. That we don't have to worry
Yeah,
too much about. Like, admittedly, I still work in journals, so I do have to interact with scientists a lot. But I we don't have to guard our careers as much. So I can say some real theoretically, I could say some really horrible stuff about people, but I won't because that'd be rude and not really good. Like, not good. Like watching for viewers. I don't think this. It's not.
yeah, yeah. I mean, we're trying to understand,
Yeah,
right? Like if in the journey of understanding we come harshly on a topic, it's only because like we're trying to right pry apart
yeah.
things so that like we can grasp
Because
them.
like we care
Yeah,
about.
we're not trying to be yeah, we're not being antagonistic just
Yeah,
because.
well,
Although sometimes it's a bit fun to show like a certain mistake. There's like some joy in
oh yeah,
that.
there is some joy. Almost like that point where because again that's almost grasping the I found something new and interesting that no one else has spotted apparently. Look, they made a mistake. Like
Yes, yes. I mean that ties into my role, right? I'm a editor for like these pharmaceutical ads, right? Like part of the enjoyment of my job sometimes is being like, I told you, you're not supposed to say those things. Like this is not done, this is not said right. Like this is too, we're stretching this too much.
oh
And it feels satisfying to catch something like that,
oh yeah,
right?
working in research integrity again, you get that same thing. Like I caught science crime. They're trying to fake their data.
Yeah.
It's a real thrill to doing that. I really I mean, the only problem is like having to deal with the aftermath of it because again, that's much more. You're dealing with real people with real feelings, and issues of
Sure.
research integrity can get very challenged, and you have to be very. Once you finding something is the exciting part. Dealing with the aftermath is the really awful, like hard work.
Right. Right. So, well, both these papers they're already published,
Yeah.
and so they've been through peer review. We'd be looking at stuff that like other people have said would be good to learn from.
hmm So
I don't know. I guess I want to go in the direction of the insectivorous one. Now, just knowing that this is like a lacuna of stuff that we haven't talked about, I just want to broaden us out and talk about something
do you
weird.
want? Yeah, I'm with you on that. Let's get weird with this. Let's talk about insectivorous
Yeah,
bats. People will be going
yeah, and MERS.
and MERS. Yeah. So So yeah, your as a break from your usual COVID-19 coverage, we're going to talk about MERS in bats.
Yeah, we're gonna talk about MERS, and the emergence of new
Yeah.
viruses. So I mean, so this is also related to some news that I also saw again looking at the backlinks of This Week in Virology. But like there was a big, so oh geez, so I'm in the States, and like the leader of the States has like a particular worldview, and that worldview like uh, led him to say that like he was going to cut funding towards like Chinese related research projects, and specifically it was there's a project about surveillance, surveilling for emerging diseases, right? So like this is an emerging viral disease. They surveil for them. Yeah, and like a big group got cut funding. They're called like the Eco Alliance or something like
Right.
that. And that grant ended. But like this is the type of work that's in that line, right?
Yeah.
Where we're talking about like this is stuff where it's like we want to know like what the possibility is in the future for something like this to come out. We want answers to questions that will help us, do the right social controls and to identify hotspots.
Yeah, and eco surveillance is so important because, like, again, like looking at what's because people might think that diseases going on in animals they aren't too important until they spill over into humans, and there
Right,
have been
right, exactly.
like some spillover events coming from China, like SARS, and that again
Yeah,
are quite important for us to keep a track of, and
yeah, yeah, yeah.
and
And so again, so like this paper is it's not the surveillance, but it's like a bit of the questions about the mechanism, right? The mechanism of that spillover,
right,
a way that it could happen. Yeah, which could inform where we do surveillance or like how we do
yeah.
surveillance. Um, yeah.
I mean,
Okay.
yeah, that sounds like a paper. Yeah, so we're going. So the title of the paper is
Selection of viral variants during persistent infection of insectivorous bat cells with Middle East respiratory syndrome coronavirus.
right.
Scientific reports uh, from May 2020. Oh, no, April 29th,
Yeah.
2020.
Okay.
Okay, I'll put it up on the website.
Yeah.
And it
I'll
was it was great to do the selection with the viewer
Yeah, great
audience.
to do with the audience. I'll put the links in the description once this stream is finished, so you get to see the new paper plus a couple of the other papers that we discussed. I think. Yeah,
Yeah, absolutely.
yeah. All right, but
All
it's
right.
been great hanging out with you. Great talking about microbiology, and
Yep.
look forward to next week.
Au revoir.
Au revoir.